Please use the link below to get the notes, questions and other videos. Don't forget to subscribe, like, and share to others for our daily new videos.
Yeah, welcome back, guys. Welcome back. Today you're going to be looking at what we call the process of meiosis. So we saw the introduction of meiosis, and today we will be looking at the detailed process of meiosis, and where most questions come from in examination. Yeah, don't forget that we say that in this video we will be having some gifts for those people who are watching. The process of meiosis. Don't forget that we say that meiosis is the process whereby one cell divides to form 4 daughter cells. These cells are genetically different from each other and from the parent. The process of meiosis, we say that a type of cell division that results in 4 daughter cells, each cell with half the chromosome number to the parent. So meiosis is a continuous process. It's a continuous process, but it's divided into different phases. And events. So the events, for example, the first event is what we call the interphase. Interphase is not part of meiosis. You must know that interphase is not part of meiosis. It's just a growth phase, because meiosis is regarded as a reproductive process in the reproductive cells, which are determining the cells which you call the gametes. So it includes DNA replication. That's where DNA replication takes place. Remember, when you're talking about DNA, we asked a question: where does it occur? We said in the nucleus. When does it occur? It occurs before the cell divides, or it occurs during interphase. So when you say before the cell divides, it means that interphase is not regarded as a phase of meiosis. It's just a growth phase. It's like saying that you give birth to a baby, and then a baby immediately cannot give birth to another baby. Needs some time for this baby to grow, so that now once the baby grows, then is ready to reproduce. When the cell divides and then goes to the last phase, it does not divide immediately. No, it requires some time for it to grow. So during that process of growth is what we call interphase. And the things which occur in that phase, some of them is DNA replication, doubling of the DNA. Chromosomes which are single-stranded, they become double. We saw it that we have what we call the unreplicated chromosomes. When they double, they become the replicated chromosomes. So when the chromosome has one thread, then we call it unreplicated chromosome, or what you call single-stranded chromosome. But when it becomes double, as we said, as you see it here, it becomes what it is called the replicated chromosomes, or double-stranded chromosome. So each chromosome will consist of 2 chromatids joined together by a centromere. So a chromosome, it must have a chromatid. It must have a centromere joining it. If it is replicated chromosome, it will have 2 chromatids, and these chromatids must be joined together by a centromere. Centromere is the middle part of the chromosome. Then DNA replication. What happens in DNA replication? DNA replication helps to double the genetic material, to double the chromosome number. As I said, that we don't say to increase the chromosome number. As we ask a question, it increases the chromosome number from what to what. So we need to say that it doubles the chromosome number or genetic material. So the material, so that genetic material, so that it can be shared equally by the new cells arising from the cell division. It means that now these cells, the new cells which are going to be formed, they are going to have the content. They are going to have the content. They're going to have the chromosomes. They are going to have other cell organelles equally, shared equally, because each cell must have this content. So if it doesn't have, then it means that it will be empty, and then it won't be able to survive. So it is the function of interphase, the growth phase, to bring about the doubling of these organelles and the doubling of the genetic material, so that each cell, each new cell, has a equal amount of the genetic material and also the cell organelles. If we have said that interphase is not the part of meiosis, then what are the phases of meiosis? Number 1, we have what called the prophase. Number 2, we have what called the metaphase. Number 3, we have what called the anaphase. Number 4, we have what the telophase. And then we go to prophase 2, prophase, metaphase 2, anaphase 2, telophase 2. So we are talking about prophase, metaphase, anaphase. What is it? What does it mean? Prophase means preparation. Pro means preparation. P versus P. Then you have metaphase versus middle. M versus M. And then anaphase versus apart. A versus A. And then T means telophase, T versus T. So even in prophase 2, metaphase 2, anaphase 2, and then telophase 2. So we form if we take the first letter, we form what we call IPMAT. This is in different color. It means that this interphase is not part of meiosis. That's why we don't have interphase 1 and interphase 2. We only have interphase. So it means that it's not a part of meiosis. So the process of meiosis begin with prophase, metaphase, anaphase, and telophase, whereby we say that this is preparation, middle, apart, and then they have reached the what? The terminal. What are those things which we are talking in middle, apart, terminal? What are they? We are talking about the chromosome, as you see them here. So this is trying to show you exactly what is happening. So it means that we start with one cell, the one cell divide from 2 cells, and then these 2 cells divide from 4 cells. But actually, what is really happening for this cell to divide is what you're going to look at in the details of each phase. Let's look at meiosis 1. Meiosis is divided into 2 phases, into 2 divisions, not phases. The first meiotic division and the second meiotic division. So in the first meiotic division, it will have prophase 1, metaphase 1, anaphase 1, telophase 1. And then the second meiotic division will have prophase 2, metaphase 2, anaphase 2, and then telophase 2. So the first meiosis, this is called reduction division, whereby when you talk about reduction division, you start with 46 chromosomes. At the end of meiosis 1, you end up with the 23 chromosomes in each cell. Hence, we call it reduction division. This division results in the reduction of the number of chromosomes from 46 to 23 in each cell. So the resultant gamete are haploid. It means that they have one set of chromosomes. From chromosome number 1 to chromosome number 23, they are not paired. It means that they are single haploid, half. It means that the cell has 23 chromosomes, not 23 pairs of chromosomes. All right. Let's look at the phases of meiosis. The first phase is prophase, which means preparation. The cell is preparing for whatever is going to be doing. So the first thing is chromatin network. We say that chromatin network means that entangled DNA. DNA is in form. We don't have a direction when we look at it. We don't know where it starts or we don't know where it ends. So it is chromatin network. When it condenses, it becomes thick. When it becomes thick, it forms what we call the chromosome. So the chromatin network condense. When they condense, it means that now that it has formed a chromosome. Now these chromosomes, the chromosome which look alike, homo, which means the same, logos, which means structure. So it means that chromosomes which look alike, they are going to come together. That's why you're saying that homologous chromosomes are formed. It means that the chromosomes which look alike, they come together. When these homologous chromosomes, the way you see it here, when they come together, they cross over. What do they use to cross? They cross using the chromatids. The one chromatid of one homologous chromosome crosses with another chromatid of another homologous chromosome, and then the process continues. So the spindle thread starts to form. It means that now the spindle fiber, they start to form, and then once they start to form, the nuclear membrane starts to disappear. Let's look at the diagram here. So you see that here, the chromosome, they condense, the homologous chromosome. You see this one is short, this one is long. So this short cannot go with the long one. No, the short goes as with the short, the long one goes with the long one. That's the principle. these chromosomes, they come together, and then the chromatids, they cross each other. When they cross each other, we call it crossing over. That's why I'm saying that crossing over takes place. And then the point where they cross over, we call it chiasma. We're going to look at this in detail. And then now these spindle, they start, the centrioles, they start to move to opposite poles. As they move, yes, as they move, the spindle starts to form. So once you see something like this, they have crossed like this, then you just know that yes, this is prophase. Why? Because the chromosomes are crossing over. Homologous chromosomes are crossing over. We don't have crossing over anywhere else except in prophase 1. Even not in prophase 2. We only have it in prophase 1. Why in prophase 1? Because the chromosomes are still in homologous pairs. Let's look at the process of crossing over. So we have seen that the homologous chromosome, the chromosomes which look alike, they're going to line together. When they line together, one chromatid of each chromosome overlap. So it means that they cross each other. When they overlap, these chromatids, they must overlap based on the homologous pattern. For example, it means that the one chromatid from one homologous chromosome and another chromatid from another homologous chromosome, they cross over. And then when they overlap and they cross over, it means that they cross over at a point called chiasma. And then crossover takes place at a point called chiasma. If it is one, called chiasma. If there are many, we call them chiasmata. So when we say chiasma or chiasma, depending on the kind of English you are using, then chiasma or chiasma is a point where these two homologous chromatids cross over during prophase 1. And then now the chromosomes separate. So it means that now they have formed different chromosomes. I always ask this question to different people. Are you sure your father is your father? Why am I asking you that? You don't resemble him. Actually, you don't even resemble your mother. You walk, you go somewhere, and then you find out that you resemble someone. Why? And they ask you that, hey, when you resemble someone, I know that. Why? Basically, he might be your father. But because of crossing over, this made to the exchange of the genetic material. If you came from the same father, same mother, you breastfed on the same breast of your mother, why don't you look alike? It means that this crossing over, this process of meiosis helps in bringing about variation among organisms. So it is a very important process to make humans, individuals, different from each other. And then this helps in the survival. If all of us, we are the same, then it means that we won't be able to survive. Imagine everyone is rich. Do you think that there will be someone who is working for another one? Everyone has money. Do you think I would have taught you this? No, because you don't need this. No one would have been watching. You don't need this. You have your money. What else do you want? But because there is difference among these organisms, among us, then it brings about what we call competition. Hence, different people will work differently and in different environmental conditions. So that's the purpose of meiosis, to bring about variation among organisms of the same species. All right, let's continue with the process of crossing over. So these differences is being brought about by crossing over. Imagine a father producing over 100 million sperm for a single ejaculation. And then this sperm, each sperm is different from each other. What made them to be different from each other is the process of crossing over, because it forms different combination of chromosomes. All right, let's look at. So we're saying that you have one homologous chromosome and another homologous chromosome. They come together. Yes, when they come together, they overlap. The homologous chromatid overlap at a point called chiasma. Yes, if you look at ABC and then decide ABC. Yes, when they cross, now this one takes this red and then the red takes the blue. That's why you see it here. The blue takes the red and then the red will take the what? The blue. These chromosomes during crossing over, scientifically we call it a bivalent or bivalent, based on how do you write that. How do you pronounce that? Bivalent or bivalent? And then after that, they separate so that each chromosome has genetically different, each chromosome has different material from one another. Look, if you see that ABC, it started with ABC, but this is a small error. The small error was found on this chromosome. And then this one, ABC, but this one is a capital letter. The capital letter came from this blue. If you look at it here, it came from the blue. It means that the blue, the red took the blue, and then the blue took the what? The red. So because of this, it's gonna cause different gametes being formed. And then if you look at it, sometimes we can bring it in the exam like this. Homologous chromosomes, when they cross from top. So when it crosses from top, it means that you have to draw it on top. Don't draw it down. That's one of ways of losing marks if you don't show the variation correctly. So they have the chromatids cross over, and then they exchange the material or the information. And then now the chromosomes now have new combination of the genes. Guys, don't forget that I told you that there is a gift in this video. Just click that link below. It will take you to a specific page on our website. And on that page, there is a code for you to obtain that airtime. Enjoy it. When you get it, when you load it on your phone, let us know that you got it. Thank you very much. Let's continue. And then you're saying that the second phase is metaphase 1. Metaphase 1, meta means middle. It means that now the chromosomes are always in homologous pair. Homologous chromosomes randomly align along the equator of the cell in 2 rows. It means that they align along the equator in 2 rows. One from chromosome number 1 to chromosome number 23. Another one is from chromosome number 1 to chromosome number 23. That's what it means. Each chromosome is attached to the spindle thread from each pole. This is what I'm trying to say. So they align in 2 rows. You see, they align in 2 rows, and then these chromosomes are being held by the spindle. See, each chromosome is attached to the spindle thread from each other. They do this arrangement randomly. So the key point is homologous because they're in pair and then each other. You see, this one cannot come here. No, they must be together like this. Homologous chromosomes. Yes, that's the key point. And then they come here randomly. Remember, we have 23 pairs, but we're just showing you 2 pairs. Yes, they are here randomly. So it means that another one can be here. This one can be on top. So this set can be on top. This one can be down. Or this chromosome can be this side. This one can be this side. That's what we call randomly. So when they separate, we're going to form different cells. So each chromosome is attached to the spindle fiber or spindle thread from each pole. That's what we are trying to show you, that these threads are attaching the chromosomes from each pole. So after that, once they are done with this, then they start to move apart, which you call anaphase. So in anaphase, the spindle thread shorten. They try to pull. When they shorten, they pull. It's like when you are a bodybuilder, when you carry something, the muscles shorten. When they shorten, they pull. Yes, so they pull the homologous chromosome to opposite poles. And then the chromosomes, what happens to this chromosome, the homologous chromosome pair, then they do what? They separate. The chromosomes of the homologous pair, they separate. It means that now they move to opposite poles. This is what is happening. So now they move to opposite poles. And then now, when they move to opposite poles, now they reach the pole. So in telophase, each chromosome reach at the pole of the cell. And then once they reach the pole of the cell, the cytoplasm and the cell membrane divide. When the cytoplasm and the cell membrane divide, that process is called cytokinesis. We call it cytokinesis, division of the cytoplasm. And then the 2 haploid cells are formed. 2 haploid cells are what? Are formed, and then the nucleus or the nucleolus, that dense body in the nucleus, they reform. Then it reforms, and then lastly, the new nuclear membrane forms, and then now the cell appears to be a full cell. So this is what I'm trying to say. Yeah, so now we form two cells. Now these cells, cytokinesis took place, and now the nuclear membrane start to reappear, and then the cell goes back as if it is in prophase. So now when it reaches there, now each cell, each of it will undergo meiosis 2. It means that now this one is going to go under meiosis 2, even this one is going to go undergo meiosis 2, so that you form 2 here and 2 here. So when you add them, you form 4 cells. So when you go to meiosis 2, what happens? Now prophase 2, you go to prophase 2. When it's meiosis 2, prophase, it becomes prophase 2. The 2 must be there. If you don't write it there, we won't give you a mark because we don't know which prophase is it. So prophase 2, what happens? The nucleolus, remember we said that it appeared, reappeared. So now the nucleolus and the nuclear membrane, they disappear again. But now here we don't have crossing over. So the new spindle fibers are formed. Yes, they start to form, and then the chromosome become visible. It means that they condense and then it becomes visible. And then now, once they become visible, now it's ready to undergo metaphase. They go to the middle of the chromosome, the cell. So now you see it here. Now it is forming like this. And then now they are ready to move this thing to attach themselves to the chromosome. But now we don't have homologous chromosomes. We have chromosomes which are not homologous. It means that crossing over cannot take place because it already took place before. Yes. So now in metaphase 2, the individual chromosome line up here. Individual or one chromosome align along the equator singly or individually. So the other side, we say that chromosomes, they align along the equator in homologous pair and randomly. Here they align along the equator singly or individually. So you have to know the difference. So sometimes you ask you the difference in meiosis 1 and meiosis 2. You can use the process to differentiate between the two. So at the equator of the cell, with the centromere attaching the spindle fiber, or centromere attached to the spindle fiber. And then now, this is the picture. If you rotate it, you will see that these chromosomes are in the middle. You see, they're in the middle. If this is the pole and this is another pole, they are in the middle. So this one becomes the equator. Remember, I said that equator is an imaginary line. It's an imaginary line. It's a line which we just imagine, but the line is not there. It's a imaginary line which divide the cell into 2 equal parts. It's the same thing, equator of the Earth. Imaginary line running from east to west and dividing the Earth into 2 equal parts. We're just imagining, but the line in actual sense is not there. We just imagine that that's the center of the Earth. So we just imagine that's the center of the cell. So in anaphase, once they're in the middle, they have to separate to opposite poles. So in anaphase 2, you have spindle fibers start to form. So the spindle fibers, they contract. When they contract, it means that now they pull the chromosomes apart. But now because they are single chromosomes, they are not homologous. Now it's the chromatids which are going to separate. Remember, chromosome is divided into 2 chromatids. Now the one chromatid is going to go one side and another chromatid is going to go to another side of the cell. So this is what I'm saying, that now the chromatids move to opposite. It was together here. Now this one pulls it this side and this one pulls this side. Check the direction of it's not facing this way, it's facing that way, because the force of pulling is coming from this side. So that's why it's supposed to be like that. When you're drawing, make sure that you take caution when you're drawing it so that you draw it properly. And check about the shadings. Also, the shadings are very important when you are doing it. The daughter chromosomes reach the poles. Once in telophase 2, the daughter chromosomes reach the poles. When they reach the poles, we don't call them chromatids anymore. We call them chromosomes. Actually, that's what we call single-stranded chromosomes or unreplicated chromosomes. So it's supposed to get time so that this time allows these chromosomes to grow so that now it becomes double-stranded again. That's why we need what you call interphase. So new nucleus then forms. It means that the nucleolus also come back. It means that it reforms. Then the cell membrane of each cell constrict, and then the process of cytokinesis occurs. When the cytoplasm divides, now the process of cytokinesis occurs, and then for haploid, it must be haploid. daughter cells are formed. And then now each daughter cell have the chromosome number to that of the parent. So that's why you're saying that meiosis is the process whereby one cell divide to form 4 daughter cells which are haploid or which have half the chromosome number to that of the parent. And these chromosomes are genetically different because of the crossing over which took place. So the daughter cells are genetically different, as I've just said, and then these are the cells which will form. We form 4 daughter cells, and then you see, we don't call these ones chromatids, we call them single-stranded chromosomes. Watch out this before you write your SBA. So the importance of meiosis. What are some of the importance of meiosis? Let's look at the importance of meiosis. Shall start from there with the importance of meiosis. M.Saidi was giving me the lesson. Thank you very much.